Hepatitis B is a major DNA virus targeting the liver, causing widespread hepatotropic infections globally. When this virus fuses with a liver cell, the hepatocytes it infects. The severe liver damage associated with the disease conditions a genetic blueprint inside the cell's nucleus, a stable structure known as cccDNA. To prevent this chronic immune battle from ever taking place, we have to prepare the body before the initial exposure, using recombinant vaccine technology. By genetically instructing ordinary yeast cells to manufacture just the outer protein shell of the virus, the HBsAg antigen, we can create a completely safe subunit vaccine. The only clinically viable way to prevent severe liver inflammation is to neutralize the virus in the bloodstream, intercepting it before it can establish that permanent cccDNA blueprint in the liver. HBV locks on to the NTCP receptor on the hepatocyte. The envelope fuses, and the inner nucleocapsid travels directly into the cell's nucleus. Here, a relaxed circular DNA strand is accidentally repaired by host enzymes into a perfect, closed loop known as cccDNA. This cccDNA functions like a rogue mini-chromosome, hijacking the cell's machinery to transcribe pre-genomic RNA, the template for building new viral particles. Inside the newly formed viral capsids, this RNA is converted back into DNA through reverse transcription, a defining biological mechanism of hepatinoviruses. Because the cccDNA remains securely hidden in the nucleus as a highly stable reservoir, the hepatocyte becomes a persistent virus-producing factory. Eventually, the body's adaptive immune system detects foreign viral proteins on the cell surface. CD8+ cytotoxic T cells arrive at the liver to eliminate the threat. These T cells release inflammatory cytokines such as interferon gamma and TNF-alpha, which force the infected hepatocytes to undergo apoptosis or programmed cell death. In individuals with weaker immune responses, particularly neonates, this T-cell attack isn't aggressive enough to clear the virus, but it is persistent enough to cause continuous, low-level cellular damage, resulting in chronic infection. Decades of this ongoing inflammation replace healthy liver architecture with nodular scar tissue, a process called fibrosis, which slowly deteriorates into cirrhosis and hepatocellular carcinoma. Once cccDNA is established inside the nucleus, clearing the virus completely is nearly impossible. Medical intervention must therefore focus on blocking the virus from entering the cell in the first place. To prevent that initial entry, we need a decoy, a subunit vaccine that presents the exact physical profile of the virus to the immune system, but contains absolutely no dangerous viral DNA. Scientists isolate a single gene from the hepatitis B virus, the sequence responsible for building the viral surface antigen, HBsAg. This gene is cloned into the DNA of a host organism, typically the yeast Saccharomyces cerevisiae. The yeast reads the inserted genetic instructions and begins mass-producing the viral surface protein. The process encounters a biological hurdle. As the yeast cells manufacture HBsAg, the target proteins become tightly embedded within the yeast's own cellular membranes. We cannot simply filter the protein out. The yeast cells must be violently ruptured using high-pressure homogenization, tearing the cell membranes apart to release the trapped antigen. To detach the HBsAg from these fragmented lipid walls, biochemists introduce non-ionic detergents like Triton X100, which extract the viral proteins into a fluid solution. The target protein is now free, but it is floating in a biological soup containing thousands of unwanted yeast proteins, host DNA, and cellular debris. The next requirement is extracting pure medicine from this complex mixture. The downstream purification begins with centrifugation and microfiltration, physically spinning and straining the mixture to discard the large cellular debris. The fluid is pumped through an ion exchange column, exploiting electrical charges to trap unwanted proteins. It then enters a hydrophobic interaction column, where HBsAg binds to the walls. Finally, size exclusion chromatography isolates proteins matching the exact target size. The extraction detergents, having served their purpose, are now carefully removed from the solution through dialysis. As the pH and temperature of the buffer are adjusted, the HBsAg proteins naturally seek structural stability. Their internal disulfide bonds link together, and the proteins spontaneously self-assemble. They fold into perfect 22 nanometer spheres known as virus-like particles, or VLPs. Nucleus treatments and ultrafiltration are applied to strip away any residual host DNA and chemicals, leaving the batch highly concentrated. Through this multi-step downstream processing, the result is a purified ghost virus, a structure indistinguishable from hepatitis B on the outside, but completely empty and non-infectious on the inside. Because these purified VLPs lack viral DNA, they struggle to trigger a strong immune response on their own. To solve this, the final vaccine is adsorbed onto aluminum adjuvant, which acts as a localized irritant to wake up the immune system. Once injected, macrophages immediately swallow the viral decoys. They process the VLP, displaying HBsAg protein fragments on an MHC2 receptor. A T helper cell docks with this receptor, verifies the data, and releases chemical signals to mobilize the immune network. These signals awaken dormant B cells, prompting them to rapidly multiply and differentiate into dedicated plasma cells. These plasma cells function as biological factories, pumping out vast quantities of anti-HBs neutralizing antibodies, specifically calibrated to bind to the hepatitis B surface protein. Simultaneously, a fraction of these cells convert into memory B cells. They will patrol the bloodstream for decades, retaining the exact structural blueprint needed to defeat the virus. Clinically, if this circulating antibody concentration reaches 10 milli-international units per milliliter, the patient has achieved the protective correlate and is fully immune. If a vaccinated individual is ever exposed to the live hepatitis B virus, the secondary immune response is immediate. Long before the virus can navigate the bloodstream and find an NTCP receptor on a hepatocyte, the pre-existing anti-HBs antibodies swarm and neutralize it. Stripped of its ability to dock, the virus is harmlessly cleared. It never enters the cell, it never forms cccDNA, and the liver remains untouched. Recombinant vaccine technology does not attempt to cure a diseased liver. It uses genetic engineering to arm the immune system's memory, dismantling a lethal virus at its exact point of entry.